It is clear there was accumulating evidence that a variety of substances in the environment might well be affecting endocrine systems of everything from fish, alligators, and humans. These chemicals were coming from a variety of sources, and it was critical to find out what their real significance was. But it proved not to be as simple as this. There seemed to be a whole load of confounding variables which made any simple interpretation of the experiments very much more difficult. We're now going to look at some of these confounding variables. Some of the categories will overlap, but by the end of it we will have a general idea about what the difficulties are in interpreting these very complex effects of environmental chemicals on human and biological systems. The first question would seem very simple. The initial focus was on the fact that the environmental chemicals seem to have estrogenic activity. That was the assumption that was happening. So the question was, how active were they? How active as estrogens actually were they? Here's the structure of estradiol, the most potent natural estrogen. And here's a list of some of the substances which have been deemed to be estrogenic. But look at their structures compared to estradiol. Okay, there are a few ring structures, but really none of them look particularly like estradiol. So really, how active were they? This is representative of some of the typical data which began to appear. We're looking at the relative estrogenic activity of various compounds compared to estradiol itself. Here's estradiol, and it's been given a relative bioactivity of 1. So everything else is compared to estradiol, and here we have estriol, which is another natural estrogen, genistein, coumestrol, equol, these are all plant estrogens, a DDT chemical, here's another plant chemical, here's another plant chemical. So we have some industrial chemicals, some plant chemicals, some natural chemicals. And we can see that they're all estrogenic to some extent, but not as much as estradiol itself. But they all show some estrogenic activity. But it's important to note that this is a log scale. Relative bioactivity of estradiol is 1. That's the endogenous most active estrogen. And all the others are much less, 10-fold, 100-fold, 1,000-fold, 10,000-fold, 100,000-fold. If we plot the data on a linear scale, rather than the log scale, so here's the relative bioactivity of estradiol, still 1, but this is now a linear scale, we can see that most of the compounds don't show up at all. They are very, very weak, less than 1,000th, 10,000th, 100,000th, some of them less than a millionth of estradiol. If they're very weak, is there really a need to worry? Well, even though they're weak, one could easily envisage that they have effects at certain times or under certain conditions. If they're exposed to large amounts of weak compounds, they could have effects, or over a long period of time, and particularly at critical periods of development, when cells and tissues are making decisions about which way to go, how fast, and in what direction. This is exactly what was happening in fish. The fish were being exposed to large amounts because of the sewage treatment works, over long periods of time because it was more or less continuous, and throughout their life, including critical periods of development. So a high incidence of intersex fish in polluted water, polluted initially, they thought, by these surfactants, which broke down to these weakly estrogenic compounds, would be a reasonable explanation. In fact, it turned out that the concentration of some of these compounds wasn't always sufficient, and a real culprit in some of these studies was actually human hormones coming out of the sewage treatment works, human hormones, estradiol and estrone, the natural hormones, and ethinyl estradiol from pills. And you need to remember something about the biology of fish. Fish are exposed to these substances, whether they're the weak estrogens or the much more potent estradiol, estrone, ethinyl estradiol, which are present in very, very low concentrations. But they're exposed to all these compounds via their gills. The gills are very vascular, very thin, and with a very large surface area. They're designed to take out oxygen and exchange CO2 with water. It's not surprising that these lipophilic compounds are easily taken up as well. And having gone into the fish, they are not subject to first pass detoxification by the liver at all, because they're going into the systemic circulation. They go to the heart, pumped around the rest of the body, with little interference by breakdown from the liver until much later on. The initial focus was on the estrogenic activity of the compounds. This was because there were very sensitive assays, in vitro bioassays, in vivo assays, receptor binding assays, for anything estrogenic, because of the background of estrogens and breast cancer. But given that these chemicals look very little like estradiol, is it really reasonable to expect that they're all working just by the estrogen receptor? This section will look at through what mechanisms do the chemicals produce their effects. Let's look first at how the estrogen receptor works. It's a so-called nuclear receptor, and our knowledge of it comes from the intense studies on breast cancer. In the cell, it exists as monomers with no ligand around, but when a ligand like estradiol comes along, it binds to each of the estrogen receptors, and that causes a conformational change, and the thing dimerizes. The dimer then migrates and binds to specific DNA sequences. That's why they're called a nuclear receptor, because they bind to the DNA within the nucleus. The binding causes a conformational change, and you have the recruitment of various cofactors, which coactivators or co-repressors, which then link with the transcription machinery within the cell and turn on target gene expression. Different ligands can induce different conformational changes in the estrogen receptor monomers. It may affect whether or not they dimerize, how tightly they dimerize, and their binding to the DNA. It may also affect how many coactivators and what type of coactivators or co-repressors are recruited, and to what extent they can turn on the transcription machinery. Some ligands may be agonists, some may be antagonists. This is well illustrated by some of the early work by John Sumpter and his group. John Sumpter was studying the intersex fish, but he branched out because of this into looking at how these compounds were having the effects. And what he used were yeast cells which had put into them either the estrogen receptor or the androgen receptor, and you could look and see whether things were affecting the yeast and whether things were binding the estrogen receptor or binding the androgen receptor. And what he showed was very clearly some compounds had both estrogenic activity. Here we are, what about bisphenol A? Bisphenol A, estrogenic activity, mild, but some, and they also had anti-androgenic activity as well as estrogenic activity. And there were other ones. Here's one of the DDT metabolites, which had some estrogenic activity again, and again some anti-androgenic activity. And there were other ones which were very mixed. Here was non-alphenol, one of the surfactants, a little bit of estrogenic activity and a little bit of androgenic activity as well. Work like this, which showed that the compounds could have a range of biological activities: estrogenic, anti-estrogenic, androgenic, anti-androgenic. The concept changed from environmental estrogens to the more general one of endocrine disrupting compounds, chemicals which could interfere with endocrine systems. We know more about the estrogen receptor than we do for any of the other nuclear receptors. Humans have about 48 genes encoding nuclear receptors, and they encode for things including the receptors for steroid hormones, vitamin D receptors, retinoic acid receptor, the thyroid hormone receptors, the PPARs, the peroxisome proliferator activating receptors. And nuclear receptors probably evolved very early on in evolution, along with the development of multicellular animals, and are one of the most abundant classes of controls in terms of homeostasis, development, metabolism, reproduction, key transcription regulators. If we look at the endogenous ligands of some of the nuclear receptors, we see they come in all kinds of shapes and sizes, many with long carbon chains attached to carbon rings. This is of course very similar to the kinds of structures you get in endocrine disrupting chemicals, bisphenol A, tetrabromobisphenol, tributyl tin, octylphenol, DES. So it's perhaps not surprising that some of these compounds can interact with some of these. So endocrine disrupting compounds have the potential at least to interact with many of these nucleoreceptors, perhaps acting as agonists, antagonists, interacting with more than one, some as agonists, some as antagonists. The potential for endocrine disrupting compounds to interfere or impact on a number of nucleoreceptors certainly widened the concept of what they could do. Rather than just affecting reproductive tissues, they could affect almost anything within the body, anything in terms of metabolism, differentiation, development, as well as reproduction. Flame retardants, for example. Here we are. This is triphenol phosphate, which was present in some old flame retardants, could bind to the PPAR gamma receptor, and by doing so would modulate fat and bone differentiation, could affect obesity, and so such compounds were known as obesogens, generating obesity. And the concept could be widened further. Why just concentrate on nucleoreceptors? Surely any of the various endocrine signaling pathways might be affected by exogenous ligands. Here we have a good example. 2010 newspaper headlines started coming out about aspirin in pregnancy, making your son infertile. Typical type of headline, catches the reader's attention. Picture in this case of a pregnant lady, again catching the reader's attention, and a opening paragraph which makes you want to read more. Taking painkillers like aspirin could leave your unborn sons infertile. The newspaper headline was based on this paper. Intrauterine exposure to mild analgesics is a risk factor for the development of male reproductive disorders in humans and rats. The authors noted that in pregnant women there seemed to be an increased incidence of cryptorchidism, undescended testes, associated with early exposure in their pregnancies to things like ibuprofen and maybe aspirin as well. So here we have the testes should be coming down into the scrotum to be present at birth. The authors followed up the observations in women with studies in experimental animals. Pregnant rats were exposed for various lengths of time to ibuprofen or aspirin. The pups were delivered by caesarean section in this case, and aspects of sexual differentiation monitored, such as the anal genital distance, and the ability of the testes to produce testosterone was looked at. In other cases, rats were killed much earlier in pregnancy to look at the developing fetus, especially the reproductive tracts within the developing fetus, and to what extent the testes were able to produce testosterone and the development of the male reproductive tract inside. The results were clear. Ibuprofen and aspirin exposure was interfering with testosterone production and therefore the potential for male reproductive tract development. The interference with testosterone production was being mediated by prostaglandins, and we know that ibuprofen and aspirin suppress prostaglandin production. So a clear pathway could be established. Ibuprofen, aspirin might interfere in human pregnancy with prostaglandins, affect testosterone production, and therefore affect male reproductive tract development. The critical experiments obviously could not be done in humans themselves, but they could do it with human tissues. In 2015 they reported a very elegant study looking at the effect of paracetamol on human fetal testis material. Human fetal testis material from abortions was put under the skin of nude mice so they wouldn't reject it. The mice were exposed to paracetamol at levels which were at therapeutic levels for humans. So this is the kind of level that human fetal testes would normally see, and they monitored in the mouse blood how much testosterone was being produced. And paracetamol at therapeutic levels just for 1 week would depress testosterone production. We started off this section asking through what mechanisms might endocrine disrupting chemicals produce their effects. And it becomes obvious that, in fact, they could do it via a whole variety of different mechanisms. by interfering with hormone production, by interfering with transport in the blood, by interfering with cell surface receptors, by interfering with intracellular receptors, by interfering with metabolism. A whole variety of potential mechanisms. The next question: how are humans being exposed to any of the compounds? We can think about adults, children, pregnant ladies, the fetus, the gonads within the fetus, the developing ovaries, the developing testes, the developing germ cells. How are humans being exposed? Let's look at the most obvious one first, the oral route, in food and in drink. Certainly there is a huge potential. This was information published in March 2020 from the United States. 40,000 chemicals used in commerce today. 12,000 of them may be used in the manufacture of food contact materials, things which wrap around food, the linings of cans, etc. At least 3,221 exogenous chemicals have been measured in human blood. At least some of those are coming from these food contact materials. This is a good example, published in the Journal of the American Medical Association in 2011. It was known that the linings of tin cans, for instance containing soup, are under release bisphenol A. So the question was, to what extent is that bisphenol A getting into the human body and into the circulation? And these are some of the results. We're looking at the levels of bisphenol A either in the urine, up on this axis, or in the blood, up on this axis, against time of day along the bottom. So this is 7 o'clock in the morning and 11 o'clock. At lunchtime, the subjects had canned soup. What happens to the level of bisphenol A? Let's look at the green dots. This is what's happening in the serum. Immediately, bisphenol A goes up, and also in the urine it goes up. What happens next? It's eliminated very quickly. It comes down again, so by 4 or 5 o'clock in the afternoon, levels are almost back down to normal. So although there's a big intake of bisphenol A into the body, it is eliminated very quickly in the urine. So is this a worry or not? Levels go high, but levels go down again very quickly. So is this brief exposure to bisphenol A significant? This is a less obvious example of potential oral intake of these compounds. Flame retardant chemicals, for instance, accumulate in dust. The dust is coming from carpets, from clothing, from furniture coverings. All of these have flame retardants, very useful because they stop fires, but they contain chemicals. Here's one, triphenyl phosphate, which can interfere with some nuclear receptors and may then interfere with metabolism within the body. UK toddlers have been estimated to consume 200 milligrams of dust per day. Why? Because they're crawling around on the floor where the dust accumulates. Humans are unique in that they actually apply some of the endocrine disrupting chemicals to their skin entirely voluntarily. In sunscreens, in insect repellents, cosmetics generally, applied to the skin, some are actually designed to penetrate the skin and therefore go into the body, or to change the skin's characteristics. Another unexpected example: thermal receipt paper, eating food, hand sanitizer, bisphenol A. What's going on here? Well, some hand sanitizers contain dermal penetration enhancing chemicals. So they alter the skin's properties that can increase the absorption of compounds such as bisphenol A 100 times or more. Where is the bisphenol A coming from? Well, in some circumstances it will come from thermal receipt papers. Bisphenol A is put on the thermal receipt papers as a developer and is present in very high concentrations. So people who are working on tills, for instance, with thermal receipt papers, handling them all the time, if they're using hand sanitizers, the bisphenol A can penetrate their bodies quite easily. An obvious potential route of exposure for babies is via the breast milk. Mothers, when they're breastfeeding, will be mobilizing a lot of their fat and liberating some of the lipophilic endocrine disrupting compounds as they mobilize the fat. But those endocrine disrupting compounds can then bind to the fats within the milk and then be transferred into the baby. This is well illustrated by this study: breastfeeding, a potential excretion route for mothers, and implications for infant exposure to perfluoroalkyl acids, PFAs. These are the sort of non-stick compounds that you find in cookware. And what the study showed was that in the mothers, the longer they were breastfeeding, the more the levels of PFAs fell in the mothers. So the fat was being mobilized, the PFAs were coming out, and the PFAs were going where? Into the babies via the breast milk. This study looked at the potential exposure of babies to bisphenol A and parabens in a neonatal intensive care unit. And they found there were all kinds of different sources of parabens and bisphenol A: the dummy nipples that the babies suck on, the indwelling lines, the stopcocks attached to the indwelling lines, the film dressings which which hold the dressings in place. So the potential is there for babies from a very early on to be exposed to potential endocrine disrupting chemicals. Then there's a whole host of general environmental exposures which might exist, from exposure in the workplace to urban environments where there's a lot of traffic and smog, to areas where work is taking place in terms of solvents, to just general socializing. What's going on? There have been many studies of whether or not exposure, workplace exposure to potential endocrine disruptors actually has any effect. These are difficult studies, epidemiological studies, and often the numbers involved are not huge, but in general an increased risk of problems is associated with exposure to things like paint, solvents, adhesives, detergents, pesticides, cosmetics, industrial chemicals, and jobs at high risk which often involve these include cleaners, hairdressers, beauticians, laboratory workers, and more generally living in industrial areas close to incinerators or areas where a large amount of waste is disposed of. This is a good example of some of the problems that industrial pollution can produce. This study looked at perfluoroalkyl compounds which are widely used in industry. They're used as oil and water repellents, coatings of cookware, carpets, textiles. They've got very high thermal stability, they've got low cost, they've got low chemical reactivity, very widely used. But where they're used, or at least where they're made, there can be contamination of the local environment and the workplace. This study looked at the reproductive health of young men in one of several areas of the world where the industrial use of PFAs has caused local drinking water contamination, and this has led to the chemicals accumulating in their blood. Male high school students who had been exposed to high levels of PFAs were compared to young men who hadn't been exposed. And the results were remarkable. The exposed group had shorter penises, lower sperm counts, lower sperm mobility, reduction in anogenital distance, and had more abnormal sperm. The general take-home message then from this whole section is that humans certainly are potentially exposed to endocrine disrupting compounds, but precisely what each person is exposed to and through what route and at what time of their life is very variable. One of the controversies surrounding the whole area of endocrine disrupting compounds is to what extent very low dose exposure is important. The problem was an over-simplistic view of things. What everyone expected was that be standard dose response curves like this, where as you increase the dose of exposure to something, you start having an effect which gets bigger and bigger and bigger and then levels off. Very low doses down here wouldn't be expected to have an effect. But we know in reality biological systems may show much more complex dose response curves. Here's the typical monatomic one where the effect just simply increases with the dose, but you can have ones that go up and down and then inhibitory effects, or just simple bell-shaped curves. It really depends on the sites of action that the compounds are having and how they interact with the endogenous systems. Von Saal, working in the States, really initiated some of these controversial studies. What he did was to do experiments like this. He exposed pregnant mice. They were fed low levels of bisphenol A on days 11 to 17 of pregnancy. He allowed the mice to give birth, and he looked to see what would happen. And surprisingly, very low doses of bisphenol A seem to have effects on the male reproductive health, in this case enlarged prostates in the offspring and reduced sperm production in the adults. But controversy came because a number of other workers couldn't repeat the same results. The question about low dose effects and their significance became very heated. There were conspiracy theories. This is a table from von Saal's paper in 2006, where he looked at whether or not independent government-funded scientists, so these are likely to be scientists working in universities, when they reported on low-dose animal studies with bisphenol A, 92% of 119 papers, so 109 papers, said yes, there was harmful effects. Only 10 said there was no effect. So these independent scientists, without doubt, were saying low doses of bisphenol A were significant. But when he looked at the publications coming from the big chemical companies, he found that none of them showed any indication that there was any harm of low doses of bisphenol A. 100% of them said, no, it's bisphenol A is fine. Now the question was, are the big chemical corporations, have they got a vested interest in actually showing there was no effect? But it may not be as simple as that. The big chemical corporations obviously have a lot of money, and it's in their interest really to get things right if they can, because if they get things wrong, they're likely to be sued. They could afford to do very big studies. Government-funded scientists depended on grants. Often, many of their studies were quite small. So it wasn't just vested interests, we think, between government scientists and chemical corporations, but practical issues. A large number of small studies or a few big studies. Can we reconcile any of these differences? Well, maybe. It may be that at very low doses, any small variable in the experimental design, even if it's not obvious, may actually become critical to how the experiment works. And this was well summed up by a very good article in Nature in 2016, which is well worth looking at. Because the fact is, there are many small variables that may well be occurring in different labs or in the same lab at different times of the year even. Different strains of mice being used, different diets, some of those diets containing compounds like phytoestrogens, not all the mice being fed on the same diet. If they're on different diets, on different strains, they'll have a different gut microbiome. The precise lighting regime they're on, the type of bedding they've got, the type of cages that they live in. Many animal house cages are made of polycarbonates, which may get sterilised and maybe release things like bisphenol A. The mice tend to chew the plastic cages. So many small variables in experimental design which may override or complicate or hide or accentuate low doses of things like endocrine disrupting compounds. And the controversy about low dose effects of endocrine disrupting compounds is accentuated by poor wording or poor use of wording adopted by some experimental studies. This is a very good review of lots of studies which had used the term low dose in the title. And what did they find? We found that the low dose moniker describes exposures covering 8 to 12 orders of magnitude. The majority, 91 to 99% of them, being greater than the upper boundary of human exposure. So while they were labelled low dose, they were well above normal human exposure. This is the kind of data they came up with. This is just simply looking at in vitro experiments using bisphenol A. Human blood concentrations are somewhere down here. All of these in the white area are above human blood concentrations. Each dot represents a single paper where the term low dose had been used to describe the kind of exposure they were giving in the experiments. So almost all of them, 90% or more, were in the supraphysiological range. Only a few fell in the normal levels you'd expect in human blood. But the question of low dose exposure and their significance to humans is even more important when you consider risk assessment. Risk assessment typically looks at three things: the hazard, that is the potential of something causing a serious problem; the exposure levels, how much and how long is exposure for; and the sensitivity of the system. Is it very sensitive or is it fairly robust? And where these intersect, you have the risk area. For instance, what's the bisphenol exposure that humans really get? Well, it's difficult to look at intake precisely because it's coming from many sources. But when it gets in the body, it's rapidly metabolized. What to? Things like bisphenol glucuronide, bisphenol A sulfate. And these are water soluble and can come out in the urine, where they can be measured. And they're measured by splitting off the glucuronide or the sulfate groups, and you can then get back to bisphenol A, which you can measure. And for a long time the answer seemed to be, well, the general levels of exposure in humans were probably not that bad, not significant to worry about, given what we know about established toxicology of the compound. And the advantage of measuring what's in the urine is it measures all intake from whatever source, whether through the skin or oral, and it's cumulative, so you can look a few times a day, see what's coming out in the urine. That should represent in some ways the sum total of bisphenol intake. The trouble is, the numbers didn't always seem to add up. Using this indirect method of deconjugating the BPA glucuronide and BPA sulfate and estimating what's in the urine and using that to measure intake, it didn't seem to actually correlate what people thought about what the known intake was. When new methods started coming in quite recently, then those new methods allowed direct measurements of the compounds in the urine, and they provided a very interesting insight into what was going on. Let's look at the kind of results they got. This is looking at bisphenol A in urine samples from both men and non-pregnant women. Using the older indirect method of enzymic deconjugation, levels in men were down here at 4.38 nanograms per mil, and in women they were a bit lower, 1.94. Using the newer, much more straightforward direct method, then the levels were significantly higher, very much higher. So it indicates that the indirect values, which people had used for years to estimate human exposure to bisphenol A, were giving a gross underestimate of the real exposure. So even if we know precisely all the hazards associated with bisphenol A, which we probably don't, and the sensitivity of all the systems which can be affected, unless we have good data on the exposure, both in terms of how much and for how long each exposure really is, then it's very difficult to assess the risk of human exposure to compounds like bisphenol A.